to introduce our speaker today, Dr. Jeffrey Blanchard. He's an associate professor of biology at the University of Massachusetts at Amherst. He got his PhD from the University of Georgia in Athens. He's director of the UMass Microbiome Center and has done a lot of work in the soil of the Harvard forest and investigating climate change. But today he's going to talk about some cool exploration of the giant viruses in those soil. So very cool stuff. Thanks so much for coming down and visiting us. One of our first out-of-state speakers in a long time. Thank you so much.
Well, thank you. It feels really good to be here. This is my first trip to another scientific institution in 18 months. So it is wonderful to be here. It is wonderful to see an audience with faces. I gave, I think, 3 talks over just before Thanksgiving, all remote, all online, and you don't see anybody. You don't even know who's there. You don't have time to check the chat or do things like that. So it's good to be here, and it's so I'm looking forward to it. I am going to, so feel free. It sounds like there's a tradition of asking lots of questions. Feel free to ask during the talk. I'm fine with that. I've kind of set it up pretty broad based because I know there's virologists, there's bacteriologists, there's protostologists, there's plant people. My PhD is actually in botany, although I worked with Chlamydomonas. And so I've tried to keep it a little bit broader and not so specific on giant viruses. And also I'm sort of a virology newbie. So I'm learning as I go, and I haven't put together my sort of total virology seminar yet. I think that's coming up here soon with some of the work we're doing in the lab. And so the title of my talk is The Hidden World of Terrestrial Giant Viruses and the Unexpected Diversity of Soil Microbial Life at Harvard Forest in these experimental warming plots. And so all of my career up until about 10 years ago, I was sort of more of your laboratory molecular biologist working with model systems, model genetic systems, working in the lab with those. I did sort of precursors to RNA-seq experiments with Affymetrix chips. And so I've done a lot of genomics, a lot of transcriptomics over the years. And then about 10 years ago, oops, let's go here. This is just working. Pointer, flip. I'm pointing the wrong way. Maybe I turned it, no. It's just working. Complex environment. It's out there. Looking back, human gut microbiome, piece of cake. We know all those organisms. We can isolate it. It's still very difficult, but relatively simple. In the soil, there are literally tens of thousands of microbial species in a gram of soil. And it's really hard to fathom that. And the data was there when I started working, but I really, maybe I just didn't believe it at the time. I thought, oh, maybe this is just an artifact of PCR and things like that. There really are that many different microbes in a gram of soil. And most of these have never been cultured. There's no reference genomes to map things about. There's never going to be somewhere. There's a lot of temporal, spatial heterogeneity. Many of these cells aren't even metabolically active at any given time. There's an abundance of DNA that binds to soil particles and is fairly stable. So this is like dead DNA. So when you're extracting DNA from soil, you're not necessarily getting all live cell DNA. Not anything that I ever wanted to think about going into an experimental system. And then because of this high diversity, it's really difficult to assemble anything into any of the reads into larger contiguous regions or into genomes from the soil. They can do. So it looks like a great experimental system, huh? To jump into and understand. I think the good thing and what is somewhat attracted to me is I've got a lot of experience in genomics and bioinformatics, and I really regarded this as sort of a life challenge in terms of difficulty of complexity that I would know that would keep evolving. So from that point, it's been a really good system for me, and very, very interested to develop technology around. So I'm not going to talk about, we've done a lot of metagenomics, metatranscriptomes on it. This basically show, oops, backwards again, that yes, there are a lot of species. It's really difficult to assemble genomes, and there's a lot of temporal and spatial heterogeneity. So when you're doing things like a soil warming experiment, and you're taking a sample from one plot and comparing it to a next plot, there might be a hemlock in one of those plots or near it, and an oak tree in another one, and you're going to have a lot of differences between those soils, and yet you're trying to compare humidity controls. In spite of that, we've seen a lot of changes, and to me, in one of the sort of areas that's really come most out is in metatranscriptomics data, looking at the RNA extracted from the soil, where we can see key changes in different biogeochemical cycles, at least the genes expressed that are related to those cycles between the heated and control plots. And so I have a whole nother talk getting into all this data that I'm not going to get into it today. But one of the things that I love that sort of made me, whoops, going backwards, I am definitely more of an organismal biologist at heart. I want to see organisms. As much as I'm a molecular biologist, but I'm also, want to understand the organisms. And having to study sort of genes in pieces, which is mostly what we do in metagenomics and metatranscriptomics. We annotate and determine the function for 200 base pair fragments of DNA. It's very unsatisfying. And as much as we can see it, it's very difficult to put it in the organismal context. So after about 5 years of doing that type of research, I started looking for collaborations where we could put together longer pieces of DNA and start to put together genomes. And so one of the collaborations that I've had going for 7 or 8 years now is with New England Biolabs, which develops a lot of genomic technologies. And one of the things that we did was using PacBio sequencing, we had to develop some techniques where we could get longer fragments of DNA, because in the field they were having a lot of trouble with that. So we got, this is not really shown well here, but about 8 kb lengths of DNA that we got reads for PacBio, much better than 200 base pairs. But when we started to go to assemble that, no two pieces of those fragments assembled together. So we had about 100,000 reads in this length of distribution. And that's because the soil is so diverse that at that scale, you don't even get any assembly of these pieces of DNA together. So I was just like, oh my God, I went through your project, and and we'd have to scale it up to some sort of cost prohibitive level to really start assembling genomes together. There's still a lot of hope, but I'll sort of talk about different ways of getting at it than sort of the bulk soil metagenomics. So the other thing that I had going on at the time was several grants through the DOE, and particularly through DOE resource facilities. So you can get DOE grants, but you can also submit for funding through their sequencing centers and other technology centers. It's something that maybe some of you should think about, especially if you can relate it all to biogeochemical cycles or any sort of DOE mission area. And so it's a little bit of a sweet spot for me since we're doing biogeochemical cycles and warming. And so we get a lot of funding, particularly resources for working with them. So we want to really look at what are the active cells and isolate the active cells from the soil and sequence those cells. And so a lot of this work is done by Lauren Otillo, who is a graduate student who had a year-long fellowship working in Tanya Woke's lab at the Joint Genome Institute in California. And so in the end, what we did a number of things, but it worked, was we started with soil and we added water buffer with a little bit of detergent into it and pelleted the soil, and then took the suspension and filtered, oops, don't have a step in here, labeled with cyber green to label the DNA, and then filter through a 5 micron syringe. And then we took what came through that and we went through fluorescence activated cell sorting. So the goal was to isolate individual particle cells that could be sequenced. And we decided to, rather than doing single cell genome sequencing, we would pool 100 cells. And with the idea that when you're doing single cell genome sequencing, a lot of times a genome won't amplify because the bacterial cell won't break, or there'll be some other issue. So you get a lot of empty wells, or you get no genomes, or you get partial genomes. And so we took the strategy that if we pooled 100 cells, we could still deconvolute those genomes and assemble them well into individual high quality genomes. And so we did this with 4 samples with 90 pools each. So we had 360 wells, or 360 pools of 100 cells, particles. It's partly giving away, but you already know the title. They weren't all cells. And then basically we did a cell lysis and MDA, which is a type of PCR that's used for single cell genomics. And then we went through the process of assembling these reads back together into genomes and analyzing the genomes. And so this strategy actually worked incredibly well. When we're doing it, I'm going to call this the cell sorted pooled genomes versus the bulk metagenome. So this is just taking soil, extracting DNA from soil, and making lab-raised sequencing. And we get usually very few genome bins that are of any kind of quality doing this method. And the number of genomes we got using the cell sorted when normalized per DNA sequencing grades was in orders of magnitude higher. So some levels, this makes a lot of sense. The soil is incredibly diverse. What we're doing is we're reducing the diversity quite a bit. And by doing that, we can assemble better genomes. And that works really well. And not only there, we get more, but we also get higher quality genomes. So taking this approach was really good from this perspective. And really, we did this to create reference genomes so that we could map the metatranscriptome and metagenome data back to the reference genomes and analyze that data in that context. However, the good and the bad is we got things that were sort of typical soil genomes, but we get a lot of really weird bacterial genomes. And so we got a lot of genomes that are chlamydia. We actually at the time had about half of the chlamydia genomes in the database, even though there's a lot of human pathogens or strains in there. We had most of the breadth of diversity of chlamydia captured in our samples from a few teaspoons of soil. So it's pretty amazing what you can do. We get a lot of bacteroides, which are actually important in the soil for degrading different carbohydrates. But we got alpha bacteria are also common in the soil, but we got weird alpha bacteria that are often parasites of other bacteria. So in these different groups, that was sort of a common theme of getting these sort of intracellular bacteria that are sometimes found in amoeba like chlamydia or parasites of other bacteria. One of the cool things in doing this though is on this outer level, we had 200 high quality genomes that we further analyzed. And on the outer level, the black bars here demonstrate that these were actually new at this taxonomic level. So many are new genomes at the genus level, many even at the phyla, order, class, and even one at the phylum level. So going in and sequencing genomes from the environment, nearly everything that you get is new, and not just a new species, but a new genus, new family. So there's so much out there that we don't know, that we haven't cultured, that we haven't even been able to assemble. And it's really still the case because metagenomic assembly is still incredibly difficult to do from soil, and there's very few environmental genomes of any quality from soil. So this is really interesting. And these are just some pictures of some of these, otter ones. And you have some ideas of why we got some of these otter, particularly smaller bacteria that often feed on other bacteria. So for those of you who don't know, these are sort of mostly been discovered more recently. A lot of them are missed by classic 16S primers. And there's a whole what's called the candidate phylum radiation, many phyla bacteria that were missed by the primers as well. But some of these, attach, and feed on other bacteria. And so we think it might be that there's 2 or 3 of these feeding on a bacteria. And then by going through the amplification step that's necessary for the single cell genomics, anything that starts with 2 or 3 genome copies is going to start to win out from the PCR step. So it's one hypothesis for why we get a lot of these small unusual bacteria, but we really don't know why. So, but in this, and this is a figure that I need to update, but in the green are all the different chlamydia genomes that we got from Harvard Forest, and the rest are all the other genomes that have been sequenced through other methods at that time. So again, a lot of new diversity, a lot of new chlamydia diversity. Again, I did not think I would get chlamydia from the soil. Hopefully it's not pathogenic. We don't know that it is, but there's a lot of human pathogen related organisms, but they're not actually, and this doesn't show it. This here is a human specific clade. So a lot of times we would see these clades of the human pathogens and ours were related to them, but they weren't in those clades of those human pathogens. And again, suggesting that there's a lot of diversity out there related to this. Lindsay showed some things related to this in her data, where we've mostly studied a society of these pathogens. We don't know what else is in the environment related to it. But so this shows the genome size and the particle size of chlamydia. What we did not even think of when we started this experiment was that there are viruses that have bigger genomes and bigger particle size than some and many bacteria. And again, we were just going after soil bacteria to create a reference sort of genome data set. And one of the postdocs working in Tanya's lab had just put together a giant virus genome from the environment. As soon as he got our data, he was like, "Can I look at your data for giant viruses?" I'm like, "Sure." And this is Frederick Schultz, and he found in our data 16 new lineages of giant viruses. And so I can sort of give a whole talk on giant viruses, but basically giant viruses have particle sizes back in this sort of range. A lot of the ones that have been isolated to date are the Mimiviridae, and often their genomes can be up over 2 megabases. So a lot of them are around 1 megabase. So they're really big. They're in kind of a specific group of what's called the NCLDVs, or Nucleocytoviricota. And they were the first genome was sequenced about 20 years ago. And they were actually isolated from amoeba in cooling towers as part of Legionnaire's disease outbreaks. So Legionella grows inside of amoeba. And they had identified these particles in amoeba, but they were so big and they actually stayed gram negative that they thought they were bacteria. They didn't look like, and eventually over time they grew those up. And so really until the last 20 years, we didn't know anything about these sort of really large viruses that exist. Now remember, I was teaching general microbiology. It was like my second year of teaching at UMass when the paper came out. I think I stayed up all night making a new lecture so I could talk about it in class the next day. I'm like, look at the size of these viruses. It just sort of blew me away. Okay, never did I thought I would be working with them. And so some of these, I'm going to point out, this is the group, it's called the Mimiviridae. I don't want to get into taxonomy, but the length of some of these branches here suggest how novel these are. So these branches are equivalent to genetic distances. Like the Sylvan virus is really different than anything else that's been ever found. All of these giant viruses represented new species of viruses. None of the 16, even though they came from only four small soil samples, again, less than 1 teaspoon each, were anywhere near identical to each other. A virus species is really different than, mammalian species genetically. And so even in our very limited sampling, we got no 2 genomes that were even the same species. So again, that, if you think of the diversity in the soil, and you're getting these incredibly diverse giant viruses that are now in different families, recognized in different families. We're getting no two alike. So you think of some kind of saturation curve in terms of going after diversity, and we're on this straight line. Okay, good. And so some of these, the Hyperion virus is still the second largest virus genome known to date. And this is with a couple of huge metagenome studies that have come out in the last couple of years. It's still huge. One of the genomes has 47 tRNAs present. These are the same number of tRNAs we have in our genome that are used. They have a lot of things necessarily, everything for transcription, a lot of proteins related to translation. Really what they're missing is ribosomal RNA. I think as soon as they get ribosomal RNAs, then they'll be off on their own. Now, again, you can think of it, these genomes are, have 1,000 to 2,000 different genes, very different than influenza virus. And what are these genes doing? Most of these genes, black here shows the percentage of genes in the genome that have no known homologs in GenBank. So it's pretty rare these days to go in and take a new bacteria genome and search against GenBank and find no homologs. And that's what you get in the viral world. You get nothing. Some of it might be because these genes are evolving faster, and so they have higher substitution rates and more divergent. But a lot of them look like nothing, and so that we've ever seen. So there's a lot of interest in companies like New England Biolabs for this because they're potential sources of new enzymes and new products for doing manipulation. But it's, even as me, somebody who's worked with GenBank for decades, to see how much new sort of genetic gene-based diversity that was there just totally blew me away. It was like, how can these be so different that they don't have any homologs yet in GenBank? And then this is just more of a gene sharing network, and mostly to illustrate that the Sylvan virus and Soli virus are really different than anything that's ever been sequenced before and don't share any genes. So again, I keep saying it over and over again, just because I keep thinking this over and over again. These things are really different than anything that's out there. Nobody has seen anything like it. And we're finding all these in a few teaspoons of soil at one place at Harvard Forest. So, and it's nothing special about Harvard Forest at all. They're probably all over the world. They've just not, we don't really have the techniques to go after these yet. And we can go into our metagenomic data and look at some of the proteins that form the shell of the virus. So we can see pieces of DNA related to those shells, and a lot of diversity there and some individual genes, but very low coverage. And we certainly can't be putting together any really good genomes out of the metagenomic data. Yeah.
So now that you have some of these sequences, can you make bait capture type strategies to likely bring it out?
So we can try. So I'm going to go back. I'll talk. I don't have this in there. So that's actually one of the things that we've been working with New England Biolabs with. So we've done a couple things with that. One is we can take the genome and we can actually synthesize pieces of the genome and make what are called baits, and basically you can go into the environment and you can hybridize DNA against those sequences and try to pull out more of that genome from the environment and understand the diversity of that. And we've actually done that, and they're still analyzing the data related to experiment where we focused on the Hyperion virus, the largest of the virus, to try to pull out other things related to that. When the other things that we did was try to develop primers based on these genomes and the related genomes here. And basically, this is all within the Mimiviridae. There were no genes. I think once we got down into this set, we could develop a primer. So these don't have ribosomal RNA, so you're dealing with primers for protein sequences. But again, there's so much diversity that you can't even come close to developing a degenerate primer. And actually, we did develop them, and they didn't work very well. You have actually one group that works with giant virus developed a set of 24 nested primers to sort of pull out diversity. It's a crazy approach. But again, they're so diverse that PCR approaches for asking what's there in the bigger sense just aren't working, aren't going to work, period. But yeah, some of these pull down type experiments, we're hoping work where we got a pretty good enrichment. One of the challenges is that the levels of the giant virus DNA relative to bacterial DNA in the soil, we're talking about 1 in 1,000 genomes or something like that. And so I think we got about a 10-fold enrichment, but still didn't get a huge amount of fragments. The other thing that is really cool about giant viruses is and also not very good from a culture perspective. There are virophages that infects protists. So this is sort of a typical protist. And the virophages can integrate in the protist genome. So this is just like a bacterial prophage in a bacterial genome. And under the right conditions, they're expressed and they actually take over and use the giant virus machinery to make more virophage particles. And in this way, they actually provide protection for the host against giant virus invasion. And so we can see actually some good sized fragments of virophages in our data sets and in the fact sorted genome bins as well. And then the next question is, who are the hosts of these? This is the thing that I get the most. And so what we have, and this is, as much as I love genomics and bioinformatics, we just have DNA. That's all we have. We've got into the soil, we've isolated, even though we've isolated cells, in the end of the day, all we have are DNA sequences. And we have no idea at all who the hosts are for these giant viruses, because it's really detached from it. And our approach, of course, wouldn't be co-isolating the host with the virus. What we can do is ask from our metatranscriptomics data, who is in the soil based on 18S ribosomal RNA reads of the protists. And we talked this morning about many of these different protist groups here, the Rhizaria and the Cercozoa being really dominant in the soil. Alveolata, a lot of ciliates, amoeba, some different algae. And so what we'd like to do is really be putting these together. Who are the hosts for these giant viruses? A lot of them don't jump out as known hosts from previous work in the field. Here are some of the known hosts. This is a paper from Alex Warden's lab. Here's just showing a lot of the protist diversity, and the red dots are giant viruses that have been isolated with particular hosts. Some of these, particularly in the marine and freshwater systems, which are often really rich in protists, and they can filter large amounts of water, they get a lot of sort of really good enrichment of these type of viruses. Where in soil, it's extremely difficult to work with, and most that have been isolated from soil would have been sort of typical laboratory amoeba systems. So there's some diversity here, things like the Rhizaria, which are really prevalent in the soil. There's no known giant viruses that infect them. Many of these groups, they just haven't been studied. A lot of them, there's no isolates in the lab to work with in the first place. And again, this is some of the great work that's been done here that I became aware of. I'm like, wow, there's actually a group isolating soil protists that we can start to think of developing panels, first try a few individual ones, and then work from there. Because we just have no idea of who these hosts are. And one way to start is sort of with laboratory-based systems. So this sort of was again another sort of switch. I tend to somewhat do this in my scientific career. And I've been actually looking for this. When I started working in Harvard Forest, I didn't want to stay at the ecosystem level for my whole career. I wanted to find some system that I could use and work with to explore further. At the time, for many years, I used the bacterial system, and that it would result from a lot of this fact sorted data. But I've kind of made the decision that where I'm going forward is with these giant viruses. There's just so many interesting things about them. their unusual biology, their life sciences, even within a small clade of them, there is a tenfold variation in genome size. So how can they just sort of shrink and expand in their genomes so quickly? And why are they doing that? Is the large genomes necessary for adapting to multiple hosts rather than being specific to a single host? There's really endless questions about their function and evolution and their potential for a new source of biological mechanisms as well as enzymes. And then I love thinking about like amoebas and protists as sort of a system for systems biology, where you're studying many different trophic levels. You have the protist host, you have different bacteria that are either symbionts or some kind associated with these protists, and then you have these viruses that are also infecting the protists. So just within a sort of a single sort of simplified system here. And then we still have over half of the terrestrial giant viruses are from our one paper. And again, it's something that, you know, there's groups that are doing culturing and the other half are from culturing. There's only about 5 that have been assembled from metagenomic data, straight from the bulk metagenomic data. So you've got a pretty good leverage point and sort of to walk into a new field. That's kind of good to walk in when you something new and people actually want to collaborate with you. Again, it's just from a few teaspoons of soil. And so this is our knowledge of all of terrestrial giant viruses is from a few teaspoons of soil at Harvard Forest. Nobody's done anything similar elsewhere in the world. We got 16 genomes out of about 36,000 particles that we sorted. So you're not going to go to NSF and say, I'm going to repeat this experiment and find, 16 more giant viruses and spend the whole, amount of money on a grant on what they would call a fishing expedition. So to repeat some of this at scale is going to be really difficult to do. But we can see at low levels in the metagenomic and metatranscriptomics data from the warming plots that the viruses are present and the genes are actively transcribed. And we have prospective hard protists and virophages in our metagenomes. There's also at Harvard Forest National Ecological Observatory Network site that's generating new metagenomic data every year. And Harvard Forest is well known, and we have a unique research experiment. So a lot of these things are like, okay, I can do this. I've got some leverage here to go after and move into a new field. And a very interesting system. And so 2 years ago, I contacted Matthias Fischer, who was the first one to isolate and sequence a virophage and also works with other giant viruses, about going to his lab. He was really excited. He had a collection of protist flagellates that he wanted to try as hosts. And I brought some soil. We're going to do some inoculation experiments. And while we were doing this, oops, it's got cut off. A microscopist, Uli Meyersdorf, wanted to work with our filtrates and do some TEM imaging of the filtrates to see if she could see any viruses in the sample. And, both again are, I can't write this into a group, so you just got to kind of go and do it. So I think a great thing about being an experimental station, I think sometimes you have a lot of freedom to do those experiments in different ways. So I went over there, and some of the things that Uli got sort of captivated by this, which is always good when you're starting with Zippo money and you go, and then somebody wants to spend 3 months' time working on your data and doing things. So this is what a typical giant virus particle looks like. You get this icosahedral shape. You get the giant viruses actually have a membrane inside of it. So a lot of it, they're like a cell. And actually there's some data that they're actually doing metabolizing DNA repair activities outside of the host. So they really push our boundaries of what a cell is. And then we get images like this, where nobody's ever seen a virus that looks anything remotely like this. So it's a sort of giant virus size. You again can see the typical icosahedral, even in this outer layer, you can see the vertices that are kind of shifted, nearly perfect rotation from this inner part of the virus. You know, and this is on my lab's wall as like the most wanted. So here we're starting this sort of collection to me, which is almost like the impossible life challenge. We've got the second largest virus genome in the world. We've got the DNA for it. Of course, we don't have it. And then we've got some, and I'll show more, unusual pictures of viruses like nobody else has seen. And these are dead-end research projects. You can't do anything once you've sort of killed and made this image. And you can't really do anything with the genome except for maybe some bait and capture type stuff, but it's still really difficult to get the isolate. And that's where really putting them all together is sort of our goal. But given the diversity of protists out there and the diversity of viruses out there, the actual chance that I ever get this in culture in my lifetime, I think is pretty minuscule. So we can sort of enjoy some of the beauty that we see. Actually, a lot of the Mimiviridae have these fiber-like projections coming out of them. Nobody's seen anything like this types of capsid structures. Again, what are viruses doing having fibrils coming out of them? It's almost like that they're flagellar for motion or somehow attachment or moving through a medium. Here's sort of a Stargate structure. Sometimes this happens when giant viruses start to open up. Here is, we don't know. Again, it looks very much like a virus in its shape and size and structure. And then we have these structures coming off of it, you know, which we kind of call the earbuds coming out of almost each of the vertices. We have things like this again, which is a typical virus icosahedral size with these projections coming off. What the heck is this stuff? No, it's really too small to be any type of eukaryote. It's not a bacteria. Is it really a virus? I don't know. It's the closest thing we have. I can't say that these are actually viruses, but I don't know what else they would be. They're definitely some life form. And then again, for giant viruses to have this sort of rod shape, but again, have this sort of capsid structure here, where you can see the capsimeres along here, especially if you zoom in. This is similar to a Pandora virus. It's sort of one of the more bigger viral particles, which are a little bit more typical in shape for this family. And then we also see things that are in the virophage setting. And I could show probably 50 more images of unusual viral structures that have never been reported in the literature. And so we're going to have a paper coming out on that. And again, it's just sort of amazing, sort of the diversity of form that's there, and that sort of matches the diversity of the genetics and genomics level. And also in these images, we can see a lot of really unusual phages. This is one of probably the longest phages that have actually ever been imaged here. This last year, so one of the things that I did was, because I do bioinformatics, I ran remote projects for 14 seniors who wouldn't get research experience otherwise using this data set. Because what I've talked about is the giant virus and a little bit about bacterial data set, but we actually have a lot of really cool, interesting phages in our data and phage genomes, some of them up towards 300 kb. So some really large phage genomes that came out of doing this self-sorting as well. Probably attached on the surface of a phage, first surface of bacteria, or inside of the bacteria replicating. But again, these came up with really nice high quality genomes. And it's kind of interesting because we actually have in the same well, we'll have the bacteria, the potential bacterial hosts that we can do correlation networks on it. So that's a sort of ongoing project with the self-sorted data from the same data set. So I sort of jumped ahead there. And then we can, of course, see images of bacteria and phages. And then I wish I'd put in my weirdo small eukaryotic gallery. But, you know, you can see these, so there's sort of growing appreciation. There's a lot of protists that are sort of under 5 microns, sort of that 1 to 5 micron range. And we see a lot of these in here. And I sent them to people that I know that are experts in the field. And they just sort of like, I don't know, Jeff. I don't know what you got. It's probably eukaryote. It's not a bacteria, but we have no idea for these weird structures. This looks a little bit like kind of some ciliates, but it's really small. I don't know if there's like little microciliates or stuff that are out there. So there's a lot of diversity there. And actually, Lindsay brought up a big point of something that I hadn't done that I need to go back and do. And again, we've been remining this data over and over, and we've actually found new giant viruses in our cell sorted data here this past year doing different bioinformatic projects. But we haven't looked to say, did we capture some small eukaryotes in this process? And it would be totally missed by the way we've analyzed the data. Because usually what you do when you're analyzing this data is you look for sort of canonical bacterial marker genes that are usually single copy or conserved across bacteria. And then you judge the quality of your genome bins based on the percentage that they have and that they don't have multiple copies of them. So that's mostly how people judge the data. And there's new tools for pulling out the virus sequences that we've applied more recently, but we didn't even really think to see if any of you carry out. Question?
So what criteria are you using to discriminate what is a virus, bacteria, eukaryote from just based on size? Just based on
No, there's hallmark genes for, like bacteria are going to have 16S ribosomal RNA.
the TEM data.
Oh, the TM. No, Matthias has just recently put together a nice presentation focused on the TM data, going through it and comparing the structures to known structures. So we've actually gone through and gotten giant viruses from different collections around the world and done imaging with the same microscope and the same techniques to compare it to it. And so we don't know for certain that any of these images are giant viruses, but they're sort of in the same size range that we'd expect to see it. They don't look like anything like a bacterial structure, the way some bacteria do have some proteinaceous coatings around them, but they don't look like those. So we don't.
It could be a bacteria that has been identified before.
Right, but a lot of them have like the icosahedral shape, or we can see those capsamere icosahedrons going all around the outside of them, and those bacteria are unlikely to have things like that. Could be, it could be some bacteria that's never been discovered that has that. So we certainly don't know for sure that any of these are actually giant viruses. That is very true. Okay, and then actually the culturing worked really well. And one of the organisms that was really good as a host, we went through. So one of the ways that you can isolate giant viruses is you have a protist host, you add in your sample, in this case it was filtered soil, and then you ask, does it lyse the host? And you do the counts with a microscope. And then the ones that have low counts, you transfer to another well with a fresh culture of the host. Usually you go through at least 3 rounds of sort of lysis and growth to get some. And so I've gone through 3 rounds, went through my time over in Heidelberg, and one of the sort of star protists was an Adriamonas one, which is what is in the collection here. And so we're very interested in working with that as potential host. I was going to go back that summer and fall to Heidelberg and work with these lysates. I've decided to leave them over there, but then the pandemic happened, and so I haven't been able to return. I'm going back actually this March for 4 months, this May for 4 months to continue this work there. But I'm really interested in working with other protist hosts. It's likely that we're going to have to do some kind of match of different protist hosts to different giant viruses to pull out these. And it's just going to be an incredible amount of work to really do that. It's not something that any one lab really can take, but we can start small with certain unusual new protist hosts and try to capture giant viruses from that. So I do, oops, I forgot. So meant to merge these 2 slides. One so I could show a picture of my son from 15 years ago, covered with soil. But you just think of, soil, and a lot of people don't really appreciate the diverse world that's there. It's just really incredibly unbelievable. From bacteria that we haven't cultured that are in our genome collections, to viruses, to protists, to other organisms. We don't know what's there at all. Even, we're just sort of starting to get the tip of this sort of proverbial iceberg. And so all of our work that I've showed you with these new genomes, these new viruses, even the imaging is just from a few grams of soil at one place in the world. And so some of it is going in with a new eye, new technologies to really discover new biodiversity. But we know it's there. We've known for many, many years that it's there. And some of it's pulling it out. And this is where I have a lot of sort of marine freshwater envy, is because those groups have been able to apply these techniques really well because of the simplicity, the system, the ability to pull through, and working with soil is just incredibly difficult. So this is sort of my final slide here. Is sort of from the stories I expected the world to be sad. It was, and it's wonderful, and it is. I just didn't expect it to be so diverse. And every year I just get a greater appreciation for how diverse the world is out there, in terms of both the genetics and the different forms that are out there, how little we know of that world. So I'd like to acknowledge sort of Jerry for starting me on this trajectory, from a laboratory scientist to somebody who works with fields and starts to explore this biodiversity. Graduate students, Will Rodriguez, Lauren Otillo, Alex Touchon, and Jake Barrick, who've worked on parts of this project. Tanya, Rex, Frederick are just huge. It's nice to have a resource like JGI where you can write these exploratory grants and do discovery science. None of this stuff that I've talked about would have been possible through sort of typical USDA, NSF, DOE sort of hypothesis-driven research. And Danielle did the fact sorting. Danielle and Elizabeth did that. I didn't. Stefan provided the tree of chlamydia. This is Matthias Horn's work in Vienna, and Matthias Fischer and Uli Merst are at the Max Planck Institute in Heidelberg, and Barton Slacko is my longtime collaborator at New England Biolabs. Okay, so I won't show any more slides. So I'll take questions in different directions. We could go many ways in conversations. Yes.
So first off, great talk. So many questions. But so one thing we know about viruses is they often mispackage DNA, right? And then they've got bacterial DNA encoded, which to me might explain things like tRNA. So for the things where you're seeing like no genes with no homologs, do you think it could just be like maybe giant viruses are really bad at packaging, and then there's no environment, there's no selection of genes anymore, so they can just drift wherever they go? Then you see those genes in your transcriptomes to actually show that they're expressed, or you know they have a function, but there's unfunctional genes, so that's what they have.
Yeah, so I think there's a couple areas. I'll try to repeat the question for Zoom. So in these viruses, viruses can package pieces of DNA from the host into their viral particle. They can also incorporate pieces into their genomes as well, sort of randomly, and they may not really have any function in the virus other than just they were captured from the host. And so how do we know that that's not a large part of what we're seeing relative to being incorporated? So with the giant virus genomes, these are well assembled. A lot of these contigs are hundreds of kb. So we don't think it's not just sort of pieces that are incorporated in. There are a lot of genes that are similar to eukaryotes, and most similar to eukaryotes. And that is a good question of how much of these are functional. From our work, we see we haven't really systematically gone through the genomes and looked at what's expressed, because usually for most of our genomes, there's sort of between 0 and 5 transcripts that map to a genome. So it's not been very interesting. From people that have worked with culture giant viruses, they do see that most of the genes are expressed. And so they don't seem to be pseudogenes, of course, but there are pseudogenes there as well. And the tRNAs have also been shown to be functional and part of the virus translational system. So those aren't just sort of mispackaged, incorporated either. You know, why? We're learning more that one of the reasons why it has so many genes, it's actually the outer shell structure, and it has an incredible diversity of proteins in it. So a good number of them contribute to that and have different functions, from host attachment to different enzymatic roles. So there's a lot of the genetic diversity in giant viruses related to the structure itself. There are certainly auxiliary genes that it's using to control host metabolism. But also in these sort of viral cells, it tries to almost put together its own nucleus, where it's controlling what goes in and out of it. And there are some groups claim that there are actually protein targeting sequences to go into this sort of subviral component in it. I don't think the data is really strong, but it also makes sense how they have to have some mechanism for recruiting some of these host proteins selectively into this area. So there's many people in the field that I can recommend to give really good talks that have sort of worked through some of the culture systems in terms of it. Some beautiful work that way. Yeah.
So especially for the novel giant virus that you found, how do you discriminate between a viriphage that's infecting the giant virus and actually that the DNA is not actually from the giant virus? How do you know that it's from the virus?
Okay, so actually, so how do we know the virophages, that the DNA is from the virophage and not from the giant virus? That it's,
Right.
it's
The virophage.
okay. Yeah, so the virophages actually incorporate into the host genome. So not the host giant virus, but the protist host genome. So one way we can tell that we have pro-viral phage genomes is to see flanking regions in the host as part of that assembly. Otherwise, and for the most part, because our assemblies from the soil are so poor, we can't make that discrimination, whether they're actually from the host genome or from some active virophage particle that was going on. Yep. I thought you were going elsewhere, but I'll save the.
I have a question.
Yeah.
So you named one hypothesis about why you're getting so much intracellular stuff. There's just more cells for each, particle that you're getting.
Yep.
So I'm wondering if maybe it's also that the chromiales are really abundant in the soil, and we just always thought of them as being intracellular.
No.
Okay,
So a lot of the weirdo ones that I showed on that sort of tree, if we look at our metagenomic data, they're not the dominant organism. Some are kind of more prevalent than others. In particular, we get a lot of acidobacteria. And so what we're missing are there's filamentous bacteria in the soil. So longer sort of rods that might be filtered out. And actinomyces would fall in that category. A lot of the high GC microbes also are really difficult to break the cell walls. And so there's twofold reasons why we're not getting one of that, why we may not be getting those. One is the amplification process, the MDA is biased that was used at the time towards AT-rich genomes, amplify preferentially. And this is actually one reason these virus genomes are they're about 30% GC, so they're very AT-rich, 70% AT. And so we got really good assembly and amplification of the viral genomes. And I think that was a little bit of the artifact of, or not artifact, but bias of the PCR method. And also it's going to be biased against the GC microbes. So we're missing a lot of those that are really common in the soil as well.
So out of 100 particles per well, how many things are you actually seeing per well?
So we got about sort of 2 to 3 high quality genomes. And so these, usually a high quality genome is something that's judged to be greater than 90% complete and have less than 10% contamination. And that's kind of based on typical conserved bacterial genes. We have a lot of medium quality genomes that are about sort of the 50% level. And then we still have a fair amount of what I call over bin genomes, which is really common in the soil. So if you have multiple bacteria that are, let's just say they're 1% difference across the genome, they might still be in that same population group, but they're going to start to co-assemble and they're going to bin together because they're most of the genome binning methods are based on nucleotide frequencies, tetranucleotide frequencies. So two organisms that are sort of in the same population or species group will go together in the same bin. And so we'll get these like giant genomes with like 30 megabases that are just artifacts. So that's a big problem right now in the metagenomics field, is sort of the more you have of a particular group, the harder it is to get high quality genomes. And so that's another reason why we don't have good representative genomes for the abundant organisms that are hard to culture in the soil. But we see that problem in our metagenome data set, our sort of filtered ones. Yeah, this. Yeah, so that's a interesting question, and Lindsay might be better at this. So they might be in protist culture collections of where you have the virus. Probably the more likely thing is you're going to have the virophages in the genomes of it, because if the giant virus was present, it would be sort of lysing the culture every time you tried to grow it. So you'd really select against that. But by doing that, you might select for a virophage that can incorporate it into your protist genome. But there's still, so there's a lot of work and interest in isolating protists directly from the environment using single cell techniques and trying to capture the virus inside of that host. So that would be in some ways the perfect situation, because then you know that virus is inside that host and you put that together. That's much better than doing it in the laboratory because you've worked it out in the environment. And Alex Warden's group got lucky a couple times and had this happen with some of our marine samples, where doing actually single cell sorting to sequence the host genome of the protists, they amplified and put together high quality giant virus genomes. And then interestingly, she went back and tried to repeat that with some newer techniques, and including newer sort of the PCR-based techniques, and didn't get any giant viruses. And that's probably because the older technique was so biased towards AT-rich sequences that it preferentially amplified the giant virus rather than the host genome. But there's a lot of people in the field that would love to be doing that, but it's very much a needle in the haystack type thing of putting together these correlations. And it gets pretty messy in the lab too, of whether it's actually in the host versus attached to a part of your sample. Washington. No, and that's again because of the diversity of the soil. We don't even, so there's two levels to the question. From a pure metagenomic perspective, soil is too diverse, and so you get too few PacBio reads, even if you did, hundreds of thousands of reads at 10 kb to really help assemble anything. From the mini metagenome, from the FACS sorted samples, the PacBio doesn't work really well on these single cell methods. And so I think needs to be more technology development for like nano and PacBio to work with single cells. And I think nanopore is probably going to push that further more quickly than PacBio will. So I think that's where we'd really like to go, is with a single cell or a few cells getting high quality genomes from those using the longer read technology. Yeah, oh, sorry.
What has traveled from your location for your source sampling? What
Oh, why did I? They're in the experimental warming plot, so they work at Harvard Forest. So I was just doing this as part of understanding soil warming and climate change and microbial community response. And so that's why they're all from the same site at Harvard Forest, is because they were part of that experiment. And I've decided that I don't really need to go anywhere else because I still haven't tapped the diversity there in those experiments. So I could travel all over the world to unusual places looking for giant viruses, but I can probably go in my backyard and just sit there and mind my backyard and for the rest of my career.
Is it possible also to study the plants in those environments to see if those giant viruses are present in the plants?
In the plant itself or in the rhizosphere of the plant?
Yeah.
Yeah, so that brings up a couple issues. So are giant viruses present in in plants or in the rhizosphere of plants? So very little is known about, so in the this larger group of the Nucleocytoviricota actually includes the pox viruses. So there are some human pathogens, and those are pretty large genomes. Those are like 100 to 200 kb. So for viruses, those are really big, but they're sort of the smallest in this phylum of viruses. And some of the other sort of related families infect other animal systems as well. But in the sort of larger scale of things, the larger ones that I showed today, the only known hosts are protists. And so that doesn't mean people have looked and found sort of pieces of giant viruses in plant genome projects, and they might have been incorporated in plant genomes, but there's no evidence for active viruses. Where they are in the virosphere. So there's a number of sort of, they're more funding challenge type issues, and how do you get this type of research funded? So one is in the soil, we know almost nothing about the protists in soil. We just generally think of them as predators going around gobbling up bacteria. They're not hugely abundant. So if you sort of do the sort of funding sort of tiers, you have sort of the fungi, which dominate the upper layer of the forest soil and have been studied for a long time. The bacteria are still the new guys. I kind of do the Red Sox Yankee thing with bacteria and fungi sometimes in terms of tension in the field of which is more important and which is better. But then you have, like, I don't know, the minor city that nobody cares about in baseball, which is the protists, and every once in a while, hopefully they'll get a big hit. But it's hard to justify the ecological role of the protists because they're not very abundant. And so to sort of get funding and ask questions about it. And we really don't know, other than that they eat bacteria, what effect they have on the plants. And again, some of the work that Stephen, Lindsay, and people in the lab are doing to show some effect of protists on the plant is actually really huge, because that's sort of like the key things that, like, I need to go to funding agencies, say, yes, look, protists and also maybe giant viruses by association, but at least start with the protists, have an effect on plants. And here are the effect that they can have with or without their bacteria. We really don't know anything about that. So having those sort of initial forays that say, yes, there's effect, and this could be important, are so huge to drive forward. And so those are things that are kind of intractable for me and like my Harvard Forest experimental warming plots.
Thank you so much for coming.
Thank you all. It is so good to be in front of a real audience. It was so nice. Not that online audiences aren't real out there. I appreciate